So I hope you enjoyed the conversation with forerunners in Indian STEM. And welcome to this session. Here we'll be talking to early STEM professionals. And I think our first speaker is Dr. Pushpika.
Yes, ma'am.
Yeah. Dr. Pushpika is Assistant Professor of Biotechnology at MIT University, Madhya Pradesh. And she's going to talk about plant abiotic stress tolerance. Yeah. So,
yes, ma'am.
Yeah, I would hand over to Dr. Pushpika to talk more about it.
Thank you, ma'am, for the introduction. I will share my screen and then I will start with the presentation.
Yeah.
So my presentation is visible, ma'am?
Yes.
Right, and it is in the full presentation mode.
Yeah, you can probably put it to full screen.
It is it visible in the full screen?
It's not, it is visible, but not in the full screen. But yeah, I guess you can.
I have started sharing my screen and now it is in full screen.
Okay.
Is it okay, ma'am?
Yeah, I guess this is fine. Although yeah, it's fine, I guess. We also have time limit, yeah.
Okay. Good evening, ma'am. Once again, I am Dr. Pushpika Udawat, Assistant Professor at the MIT Institute of Biotechnology, MIT University, Madhya Pradesh. So my presentation, it is entitled with the proteins with domain of unknown function. They are a novel candidate for plant abiotic stress tolerance. There is a plant, an extreme halophyte, that is Salicornia brachiata. It completes its normal life cycle at a salt concentration of more than 250 molar. So, due to its ability to complete its life cycle at such a high salt concentration, we have collected this plant from the coastal regions of Gujarat on the borders of the Arabian Sea, and then the plant was grown under the laboratory conditions for further functional analysis and for further studies. So the present study, it was undertaken for cloning and isolation, cloning and characterization of an salt responsive gene. We named it as a Salicornia brachiata salt responsive gene 2. Okay, so the first objective was the isolation of this full length gene from Salicornia, then doing its transcript profiling using the quantitative real time PCR analysis. Then by studying its in vivo localization to have a knowledge about its location in the cell, and then making this gene a full length one so that it can be transformed into the plant model plant, that is Nicotiana tabacum, by agrobacterium tumefaciens mediated transformation, and finally confirmation of the transgenic lines by means of the gene specific primers or by the vector specific primer for analyzing the lines for their salt tolerance, and then the future aspect. So first, initially, what we did was we have developed the
Dr. Pushpika, sorry to interrupt. Actually, your
yes,
slides
ma'am.
are not moving. We still see the first slide.
Now?
Yes, now it's better. If you like, you can click on the bottom right, 3 bars that you see. You can click and go on to full screen. So that is an option if you can. Where there is 62%, you see that near that there is this symbol. If you can click it. Or you can just, yeah, you can just continue, I guess. Yeah. Yeah, sorry to interrupt you.
No, ma'am, it's okay. Okay, so then what we did was, so initially the EST database of this plant, from this plant was generated and it was submitted to the NCBI. Okay, so because we had ESTs, the number were more than 945. So because this gene sequence, which was specifically targeted to make it full length and then to study its transcript profiling, the sequence, it was retrieved from the NCBI database, it was then made full length, and then its transcript analysis was done. So for this study, for this particular objective, because initially I started with a set of 25 ESTs, which are considered to be salt responsive, but their function is not exactly known. So the Salicornia brachiata, the plant sample was collected from the coastal regions. It was grown in the pot, that is in the greenhouse condition, and then under the plant tissue culture laboratory, wherein the plant, they were grown under hydroponic conditions. Herein you can see that this plant, it was grown. Then the RNA was extracted, it was purified, and then the transcript profile or the relative fold expression, it was studied. And I found that there were several genes which showed upregulation under the salinity stress of 500 millimolar, which was given for 24 hours. And this gene, it was specifically targeted because it showed a very high expression, very high level of transcript. It got accumulated under the salt stress. So moving ahead, this EST sequence, it has an accession number of this, and having the EST length was 486 base pair. So this is how, this is why I targeted specifically this gene sequence, because it showed a very high expression, fold expression under the salinity stress. After having a knowledge about its transcript level, okay, so then having this studied, thereafter the full length gene was isolated by 3' rapid amplification of 3 cDNA ends and 5' rapid amplification of cDNA ends. Herein we can see that the PCR product, it was tailed, and then the nested PCR was performed, colony PCR was performed, plasmid was isolated, and it was subsequently sequenced. And on the basis of the homology search, I found that the gene is showing 100% homology from the sequence that I received from my this subsequent experiment. Moving ahead, after having made this gene a full length one, the in silico analysis, it was done. So for this in silico analysis, the protein, the nucleotide sequence, it was subjected to the basic local alignment sequence tool, wherein I found that it showed homology to the proteins which are completely uncharacterized, proteins which are completely unknown, or they are novel. Because there is a difference between the genes, or the genes whose sequence is available, we call them as unknown genes, but whose sequence is also not available, we call them as a novel gene. Therefore, this gene was specifically targeted to make it full length. Then after making this gene a full length one, I found that the total length is 1262 base pair, but the entire gene does not code for the protein. The actual coding region is 753 base pair, and it codes for a protein of 250 amino acid, having a 5' untranslated region of 294 base pair and 3' untranslated region of 215 base pair. So this is the exact sequence information of the gene, that is the salt responsive gene 2. It was named 2 because initially one gene is already isolated. It has been the sequence is submitted to the NCBI full length one, and the work is published in the Frontiers in Plant Sciences. So herein I have done the domain analysis using the NCBI tool, that is conserved domain database analysis, and it specifically categorized the deduced protein with the domain of unknown function 581 superfamily. And these proteins of unknown function, that is the domain of unknown function 581 family, they are completely uncharacterized protein, but they have a domain which resemble to that of the zinc finger like protein. And after having done its transmembrane analysis, it was found that the protein could be, it is just an assumption, but it is not practically proven, that it could be located to the in the cell membrane as per the membrane index we observed. Then after having done its in silico analysis, the transcript profiling was further studied under different salt concentration and under different type of abiotic stresses, because we know that around 1.5 million hectares of agriculture land, they are affected by soil salinity, and salinity affect around 20% of the irrigated land. So herein, the expression analysis of this gene was studied under the varying concentration of salt starting from 0.05 molar to 0.1 molar, 0.25 molar, and 0.5, and subsequently the salt concentration was increased to 1 molar. And I found a very interesting result that as the salt concentration increased, the fold expression or the transcript level of this gene, it show higher accumulation, resembling that, giving an indication that this protein plays a crucial role in providing plant tolerance towards different type of abiotic stresses, because in nature, not specifically the salt stress is there, but there are combination of stresses that a plant face, because plant are sessile in nature, they cannot move. So therefore there are a variety of underlying mechanism, a crosstalk happens between them, and this protein, if it is characterized completely and the molecular mechanism is completely characterized, then it can be further transformed into the essential crop or our strategic crops like legumes, cereal, and they can be improved for their abiotic stress tolerance, because abiotic stress tolerance cause a huge loss in crop productivity around the globe. And we know that the world or the global population is rising. We are observing a population ballooning effect. So then in another set of experiment, the relative fold expression was studied under salt stress of 0.25 molar, that is 250 millimolar salt concentration. Desiccation stress was also given, and then heat stress was given along with the cold stress. I found that under all the three type of stresses, that is the salinity, the desiccation, and the heat stress, the transcript level increased substantially up to more than 8-fold. Herein we can see that the green bar and the red bar, it shows this. But specifically under the cold stress, this protein, it showed down regulation. The transcript, it went down. The transcript level was very much lower than that was observed in case of the salt and the desiccation stress. Therefore, in the subsequent studies, only the salt, desiccation, and the heat stresses, they were considered, because a conclusion can be drawn that this protein does not have any significant effect, or it does not provide plant tolerance toward cold stress, because cold stress beyond a certain temperature, plant also faces a stress, because the membrane, it starts to denature. Then in the subsequent studies, this gene, it was cloned under the control of a constitutive promoter, that is the 35S promoter, and this vector was Picambia 1301, and the selection marker was hygromycin, that is the antibiotic. Okay, so using this cassette, that is in frame, the gene was cloned, the cassette was prepared using these two set of restriction endonucleases, that is the BAM-H1 and the SAC1, and then the transgenic lines of tobacco, they were prepared, and the presence of the transgene, that is the gene of interest, it was identified by using the HPT2 gene marker, that is the hygromycin resistant gene, which is present in this promoter, and this gene was under the control of the constitutive promoter, that is the hygromycin 35S promoter. Moving ahead, After having made the cassette, the plant, the model organism, the Nicotiana tabacum, it was transformed by means of the agrobacterium tumefaciens and the transgenic tobacco lines, they were generated. And the presence of the transgene, it was initially checked by means of the Gus assay, and then the plant, they were raised starting from the leaf disc, the shoot were induced, roots were induced, entire plant was generated, and then it was acclimatized under the greenhouse conditions, and subsequently the transgenic lines, they were studied for different parameters like the molecular, the biochemical, the physiological, and the morphological ones. Moving ahead, these are the transgenic lines which were developed having the gene of interest, that is the gene with a domain of unknown function gene. So the probable function that this gene could play are, it may regulate metabolic processes of nitrogen compounds, processes of transport, signal transduction, developmental processes, and it could help in establishment of localization in cell. It could help in regulation of transcription, translation. It may act as a regulatory factor, or it can phosphorylate or dephosphorylate a set of proteins, or it can act as a signaling molecule. It may have a catalytic activity, cell differentiation, it may have a certain role. So all these things are, they further needs to be studied. But it is the initial level screening that I have done, and it can be, the conclusion can be drawn that it plays a significant role in plant response to abiotic stresses, not specifically the salt, but also toward different type of abiotic stresses. So it may act as a cell surface receptor. It could be involved in cell signaling, intracellular transport, or regulation of gene expression. So each one of them has to be studied in detail, so a molecular mechanism can be developed specifically for this gene, because the conserved domain has found certain activities or certain factors which are associated to the enzyme like transferases, hydrolases. Because this protein, it showed binding affinity towards DNA, it confirms that it may act as a zinc finger-like domain, and it can be a protein or a specific domain binding. It can be a transporter, it can be an actin or microtubule binding protein, or it may act as a cell receptor. It can be a transcription factor, a kinase binding, metal ion binding, organic cycle compound binding, any one of them. This gene may have a function. So the future plan for this specific study is
Dr. Pushpa, sorry to interrupt again, but your 15 minutes are up now.
okay, ma'am. So I should
you can
explain it.
You can wrap it up quickly, and then we'll go in the next session, next part where you tell us about your journey.
Okay, surely, no problem. So herein, I plan to have a genome sequence of this plant because it has a huge resources in terms of unknown, uncharacterized, or novel protein. Because this plant, it has a high industrialization potential. It has a high essential oil, that is specifically the linoleic acid. And there are several plant bioactive compounds and other secondary metabolite that could be extracted, and they may be utilized at industrial level. Industrial scale production can be done, and they can be utilized in several industries, starting from the pharmaceutical to the food industries. So this, I plan to have a genome sequencing of this plant, and there are 4 fundamental aspects that should be considered when embarking on a new genome project. That is, because the genome size of this plant is not known, the level of ploidy, whether it is a diploid, whether it is a haploid plant, whether it is heterozygous or not, what is the GC content, what is the complexity. So they all will directly affect the overall quality and cost of the genome sequencing, assembling, as well as the annotation. So this is what I plan to undertake these studies in my future research. So this is the methodology that I will follow. Herein, I will find out the heterozygosity, the complexity by either by flow cytometry, then by K-mer frequency distribution, then the third generation sequencing either by the Pacific biosciences they will be performed, so that the genome can be annotated either either in an ab initio mode or either an evidence-based prediction approach. So this may be done in the future studies. So I finally acknowledge the founder president of the MIT University, Dr. Ashok Chauhan, the chairman of the MIT University, Madhya Pradesh, Dr. Asim Chauhan, the vice chancellor, AUMP, Lieutenant General VK Sharma sir, pro vice chancellor, Professor Dr. MP Kaushik sir, head of the department, Professor Dr. Rajesh Singh Tomar, head of the institution, Professor Dr. Vikas Srivastav, Dean Research, Professor Swapnil Rai, officiating Director General, Professor A. Chakrabarti, Manager Recruitment and Scientific Alliance, Ms. Neha Puri, Manager Scientific Alliance, Ms. Shefali Kashyap, Assistant Manager Avinash Chauhan, and all the staff members of the AIB. So this session is open for question and answers. We can discuss it. Thank you for giving me this opportunity, ma'am.
Yeah, thank you for your talk, Dr. Pushpika. And like you could also explain a little bit about your journey, like how you transitioned to academia after your PhD or postdoc.
Okay. After I have done my PhD from CSIR, Central Salt and Marine Chemical Research Institute, Gujarat. Thereafter, I shifted to the Delhi University, Department of Plant Molecular Biology, South Campus. I was with Professor Akhilesh Kumar Tyagi. Therein, I was specifically working on the rice, and there were a set of specific transcription factors that we were working on. There, I was awarded with the UGC Kothari Fellowship and the UGC Women Scientist. Thereafter, I joined here. After having a break, I joined MIT University, Madhya Pradesh as assistant professor. So now I plan to have research project. I plan to have laboratory. So the academic as well as the research work, they will go all together.
Okay. Thank you so much, Dr. Pushpika. And if anyone has any questions, they can please ask or put it in the chat box. So I guess there are no questions.
Okay, ma'am.
Okay.
I should stop sharing my screen?
Yeah, I guess you can stop. Yeah.
Okay. Thank you, ma'am.
Thank you. So our next speaker is Dr. Dheeraj Bhatia. Dr. Dheeraj Bhatia is assistant professor. Yeah. Hello, Dr. Dheeraj Bhatia is assistant professor biological engineering at IIT Gandhinagar, and he's going to tell us about DNA nano devices. So yeah, over to you, Dr. Bhatia.
Yes, thank you, Richa. Can you see my screen, and am I audible properly?
Yes. Yeah.
Okay. Thanks a lot to all the Sci-ROI committee for giving this opportunity to once again connect with you. I think from IIT Gandhinagar, this is my third year I have been connected with you all, and it's really a pleasure. We have been really happy to say that from last 2 years, IIT Gandhinagar has been actively working with Sci-ROI. In fact, one of our newest faculty member, Dr. Subramanian Swaminathan, who is joining us, he was selected through the Sci-ROI campaign, which is really very good. So we are also equally thankful. Yeah, we are also equally thankful to you all. Before I start my talk, I have a small disclaimer. Actually, I'm outside. I'm actually on a road. My car, the laptop is on my car, and there might be some background noise. So I request apologies if there's some background noise due to some unforeseen circumstances. I'm traveling, but giving a talk from a roadside on a car. So just please bear if some background noise is there. Okay, so my lab is based in, so Yes, I can start, right, Richa?
Yes, yes. Thank you so much for
Great.
giving this talk.
Yeah. So my laboratory is based in IIT Gandhinagar in biological engineering, where we apply the principles of biology and understanding the science of stem cells and cancer cells, and then applying those principles using nanotechnology, especially DNA nanotechnology, to program the receptor organization on the membrane, programming the signals which are passed from membrane to nucleus, and thereby studying the processes like cellular differentiation in 2D, 3D, and this will have implications more in regenerative therapeutics. So let me quickly begin with with a small introduction. Like, we all are familiar with DNA, but in my lab, or especially if you ask a nanotechnology or a polymer engineer, we don't look at DNA as the biological material. Like, we look at DNA as a polymer which can store information in terms of the Watson-Crick base pairing, and it has a very nice persistence length. Like, a double stranded DNA can act as a rigid rod up to the distances of 50 nanometers. And using these two properties of the DNA, we kind of explore the self-assembly principles at the nanoscale, where we can make one-dimensional, two-dimensional, and three-dimensional nano devices from the DNA. These are all called as the static devices, which means once it's formed, it will remain as it is in the test tube. And on the other hand, you can also put the conformational flexibility in the DNA, where you can induce the conformational changes or the structural changes in the DNA in response to external stimuli. And this can lead to the development of DNA computational based devices, where you can make DNA robotics, walkers, sensors. Especially, I'm sure many of you must be aware with the work of Professor Yamuna Krishnan at U Chicago, where she has made an array of DNA-based sensors, where you can now sense small ions to molecules to metabolites, and then apply them not only in the healthcare to understand the disease, but also to kind of program the chemical language in which the organelles talk to each other, or the biological processes are happening, right? And my work over last 15 years, right from the PhD postdoc at IIT Gandhinagar, has been focused mostly on this class of DNA nano devices, which are this three-dimensional DNA polyhedra or nano cages. Now, when I show this slide to all the students or the audience, the first question which comes in their mind is that what can you do with these devices? Like, if you see this DNA-based nano devices, they have two very key properties. Like, they have very well-defined internal volume, which can now be explored to encapsulate proteins, therapeutics, nanoparticles, so on and so forth. And not only that, they have a very well-defined 3D structure, which can be programmed chemically by coupling them to peptides, small molecules, proteins, so on and so forth, for the biological targeting. So just once again, I'll switch off my camera just to be. Just one. Give me a second. I'll be back.
Yes, no worries.
Yeah. Yeah. Is my screen is visible, right? And the sound is okay.
Yes.
Okay. So in my lab at IIT Gandhinagar, we use the like, we use a combinatorial approach, where we make different DNA-based nano devices using origami and modular approaches, and we also explore the functional properties of the DNA nano devices, like coupling them to small molecules, peptides, antibodies for targeted delivery, and then we apply them to biological systems. Like, can they sense the receptor clustering in the membrane? Can they mark specific endocytic pathways, so on and so forth? And these are some of the publications from the lab at IIT Gandhinagar. We are only 3 years old at IIT Gandhinagar, but thankfully the projects have taken off very nicely. The lab has come up very quickly. So in today's talk, I will show you one area of research where in my lab, where we explore more DNA nanotechnology to understand the principles of membrane trafficking. Now, if you see the plasma membrane of the cells, at, at any moment of time and space, in the plasma membrane, there are many processes happening, or the key endocytic pathways which are happening at the plasma membrane can be classified into mostly clathrin-mediated endocytosis, where the cytoplasmic protein called as clathrin forms the coat at the pit where the ligands are bound to the receptor, and then it is cut by a protein called as dynamin, and then the molecules get endocytosed inside the cell. This has been the classical pathway which has been studied over the years. And what was observed in the late 90s is that when you knock the pathway out, like when you block the clathrin pathway by either small molecules or siRNA or so on and so forth, still a bulk of molecules get endocytosed inside the cell. And some of them, they get endocytosed in uncompromised efficiency. And this gave rise to the concept of non-clathrin pathways or the clathrin-independent pathways, such as the one mediated by the actin or the caveolae at the muscles. And our lab has studied very largely the processes of clathrin-independent pathway as well as clathrin-mediated pathway using a lot of biological principles. And we started to ask, when you have small DNA-based cages or small ligands of different sizes, is there a preference in terms of binding to the receptors and getting internalized? And if yes, what does it lead to? What are the implications in the cells? So we started by synthesizing DNA nano cages of different geometries and different sizes. So here I have shown you, on this slide, the DNA cages of different geometries, like the tetrahedron, a cube, icosahedron, and the buckyball, which are all made by small pieces of DNA. These are small DNA strands which anneal together in test tube. And when you do a small PCR-based reaction, they lead to the formation of those DNA cages, which can be easily characterized by the gel electrophoresis, dynamic light scattering DLS, and the AFM, atomic force microscopy, which tells you the formation of those DNA cages of particular sizes and geometry. When we were able to successfully make this DNA cages, we asked the question that does the size and the geometry of this ligand plays a role in terms of getting endocytosed or internalized inside the cell. We began with very simple pyramid or a DNA-based tetrahedron of different sizes. So if you now change the dimensions of the DNA or the length of those DNA primers, you can create DNA tetrahedron of different sizes. Like you can make a small DNA tetrahedron of 10 nanometers, 15 nanometers, 25 nanometers, so on and so forth. And what we found was that there is a very, very specific size-based window where the molecules of only 10 to 15 nanometers get internalized very nicely inside the cells. Now, this has been one of the biggest challenges in the community, where if you see the gold nanoparticle-based devices or polymeric devices, nanoparticles which have been used for applications in drug delivery, people have made the nanoparticles of like 100 nanometers, which somehow get endocytosed into the cell by the mechanisms like the macropinocytosis. But efficiencies have been one of the biggest limitation in terms of successful drug delivery, immunoengineering, or bioimaging. And what we found was that instead of making the bigger nanoparticles, if you make small nanoparticles, like the 10 to 15 nanometers, they get very nicely, efficiently internalized. And then once we had the control over the size of the particles, then we started to vary the geometry of those particles. And what we found was that out of all the geometries, like the cube, pyramid, DNA tetrahedron, icosahedron, buckyball, only tetrahedron was the one which had a very high efficiency of internalization into the cells. And not only that, like the geometry, the size, and the cell type. Because what happens is, even though the composition of the membranes are same in the different tissues in the body, still what happens is the level of receptor present or the receptor expression varies from cell type to cell type. And we found that these devices had more endocytosis into cancerous cells like SUM or HEK293, and very, very less into the epithelial cells like the MEFs. And there we found that they get very nicely internalized into the breast cancer cells, in the kidney cells, and based on their geometries and sizes. The biggest question was, what is the mechanism by which these molecules get endocytosed? Like I showed you that the key pathways can be classified into clathrin-dependent and clathrin-independent pathway. And we tested all these pathways by using inhibitors. And now we are also doing the siRNA and CRISPR-based knockdown of those pathways. So the very classical inhibitor of clathrin-mediated pathway is this pit stop 2, which blocks the endocytosis of a very classical clathrin marker like the transferrin. Oh, there's a chat box here. Okay, yeah. And we have used different markers for different endocytic pathways. For example, transferrin is a very well-known marker for clathrin-mediated endocytosis, and galectin-3 is a very, very well-known and established marker for clathrin-independent endocytosis, where galectin gets endocytosed by the glycosylated receptors and actin-mediated endocytosis. And when we looked at the behavior or the uptake of DNA cages and these markers upon inhibition of the clathrin pathway, what we found was that the transferrin endocytosis was completely blocked, galactin-3 was very minimally marginally blocked, and the tetrahedron was highly blocked, the internalization, indicating that maybe the DNA cages are getting endocytosed by the clathrin-mediated endocytosis. We cross-validated our results by blocking the protein dynamin, which cuts this clathrin-mediated pits at the plasma membrane using the molecule called as dinosaur, and again the recapitulated set results indicating that the small nanoparticles mostly prefer to get endocytosed by the clathrin-mediated endocytosis. We have also used a control inhibitor, that is a lactose, which blocks the clathrin-independent endocytosis, that is the galactin-3. If you see here in this image, the galactin-3 endocytosis is completely blocked upon the usage of lactose, whereas the transferrin and the DNA endocytosis has been not affected, thus confirming the results that the DNA cages prefer the clathrin-mediated endocytosis to get internalized into the cancerous cells. Right? And this was something very, very first observations which have been done. And now we very recently received the CRISPR cells where the clathrin adapters, the CAM adapters and AP2 have been blocked. So we are getting now the results from the genome edited cells and then kind of verifying our results. And then the main question is, when these molecules go internalized into the cells, what do they do? What do they do? The answer is, when such events are happening at the plasma membrane, the not only endocytosis is happening, along with the endocytosis, it also triggers different signaling pathways like the MAP kinase, SARC kinase, probably JAK-STAT signaling, so on and so forth. And most of these pathways are related to the processes like the signal transduction to the nucleus, differentiation, and enhanced migration. And we developed in the lab a 3D invasion-based model where you can make a spheroid out of the MDMB231 negative breast cancer cells and study their invasion in 3D. So here, if you see on the right side, there is a control cells where very few control spheroids, 3D spheroids, where very minimalistic invasion is happening from the tumor into the collagen. But the moment you add DNA cages, the tetrahedron, and incubate them, you see that the invasion has been increased 3 to 4 times. Now, this is something very exciting because not only are we seeing that the cages are getting internalized, but they're also triggering the signaling processes. And this is one of the biggest advantage, that you can now kind of or neuroactivants or drug molecules to block those pathways in a very, very efficient way. So very specific studies are under the way right now. And not only we observed the processes in ex vivo in the 3D tumor models, but also in the vivo in animals. So we used a zebrafish as a model system, and if you soak the zebrafish in a pool of DNA cages, what happens is that these fishes have a very strong barrier, that is an epithelial barrier, blood-brain barrier. And it has been very challenging to overcome these barriers using the classical strategies. What we found was, again, that using the small pyramid-like DNA geometry, these cages were able to break or overcome these barriers and get very nicely internalized into those sections. So what you're observing
Dr. Bhatia, we cannot hear you anymore.
Hello?
Hello?
Yeah, is it better now?
Yes, we lost your Yeah, lost you in
Yeah,
between.
sorry.
Yeah.
Yeah. Is it better now?
Yes.
Yes. Okay. So I was saying that we have validated our results not only in the cells or in the 3D tumor models, but also in the in vivo model systems using the zebrafish brain slices, where we see that when you soak the fish in the pool of DNA cages, what happens is a small pyramid-like geometry nanoparticles, the DNA-based tetrahedron, can overcome the barriers like the blood-brain barriers, the epithelial barriers, and get successfully endocytosed into those brain components of the fish. Now, this is something very exciting, and this opens up the new arena of research that can you now specifically target them to the particular tissues, or can you now see the release of the drugs from those DNA cages in the in vivo systems. So these are some of the projects which are ongoing in the lab. And if I have to lastly summarize my work of over last 15 years, then what we have developed is DNA-based nano applications. Like we all have the smartphones in our pockets, and even if it is just one device, it can actually have many jobs. Like you can talk to the friends, attend the meetings, book the tickets, book the food, travel, so on and so forth, using different applications embedded in the same device. Similarly, what we are developing is DNA-based nano apps for on-demand applications, where my lab has expertise on three-dimensional DNA cages, where you can put nanoparticles for imaging, gold nanoparticles or magnetic nanoparticles for the purification of the brain components or any cellular component or tissue component, and then analyzing them for the proteomic content, so on and so forth. And our focus now has been moving towards the applications in neuroscience, where we are not only developing the devices with better capacities to cross the blood-brain barriers, but also to kind of develop the devices to block the progression of the neurodegenerative proteins, or can we make a sensors out of those DNA cages which can identify the early oligomers in the neurodegenerative diseases like the Alzheimer's and Parkinson's and block their progression. So these are very preliminary results in the neuroscience, but I'll be happy to discuss if anyone has specific questions. And the second branch of our research is in the translational nanomaterials, where we are again programming or developing, modifying the surface of the DNA cages with small peptide-based growth factors, which have very, very specific instruction to give to the cells. And then we were studying the stem cells differentiation and understanding, can we now make the 3D organs from these DNA-based devices. So these are very, very first results from my lab, where we are now take the human umbilical cord stem cells (UX) and group them in presence of the DNA cages. What we find is that these cages have the capacity to induce the formation of the umbilical cord like material in within few hours. So these are very, very first results from my lab, where we are now venturing into the stem cells field. We have the fully functional BSL-2 lab for the iPSCs and patient-based cell lines. And with this, I would like to thank the organizers for giving this opportunity, wonderful opportunity to interact with the colleagues through Sci-ROI. I'm thankful to IIT Gandhinagar for the support, DST for the Ramanujan Fellowship. We have grants from DPT, BRS, Haifa, and some of the industry-based projects are also ongoing in the lab. And this is the team which has done all the work. Like we have a good bunch of postdocs, PhDs, MTechs, JRFs, and still I have good openings in my lab. If somebody is interested, just drop me a word, and I'll be happy to discuss more in details. So with this, I'll stop here. I hope I'm on time.
Yes, yes, Dr. Bhatia, that was a very nice talk. Indeed, it was very interesting. And also, could you tell a little bit about your background, like
Yes.
how you transitioned to India and your journey?
Yes, yeah, absolutely, yes. So I finished my PhD from PIFR Bangalore, NCBS, and I moved to Paris. I was a postdoctoral fellow at Institute Curie, where I was studying the cancer progression, how the sialic acid content at the plasma membrane modulates the 3D invasion potential of the cancer cells in 3D and in vivo. And at the end of like 4 years, I had good number of publications, and like that was the time to take a decision. And like I started to apply actively to India. One advice which I think I always give to the people, which I also followed, is don't apply just to one or two institutes and wait for the decisions. like in India, the process is very slow. The number of applications are very high, and it takes time to process the applications. So when I decided that yes, now it's the time to move back to India, I applied to almost 30 places in India. I applied to all the CSR institutes, IITs, ICERs, and when I did that, like I got 8 interview offers. So that you take a break, come back, give the interviews, and out of it, I had like 3 offers. So I could decide which is the best place considering the family and the 2 body problems and everything. So that was a journey. It took almost more than 1.5 years after publication of the papers from the postdoc. So one advice which I always give is, of course, postdoc is a good journey. You will get your first paper very quickly, maybe in 3 years or 3.5 years. But the moment you think you're ready, please don't wait for the next big paper, because you never know, it will get delayed. Apply early if you're young. That is one of the biggest advantage. Like below 35, if you are, that's a big advantage. You can get good offers. Still, I would say situation is very good in India. A lot of institutes are looking for it, and do apply. Do get in touch. That is what I did. Like I knew people in India, so I just wrote to them. I said, "Can I come and visit you? Can I come and give a talk?" So that started build on the network. So please don't feel shy. Just write to the colleagues, even if they are not known to you. Just write to them. Many of them they'll reply and be happy to help you further.
Yeah, thank you so much for sharing your journey and for your advice, Dr. Bhatia. That was really nice. And now we're open. Yeah, and now we're open to question answer session. If somebody wants to ask a question. Actually, I
hello,
would. Yeah. Okay. Yes, Mr. Govind.
hello. I'm Govind, postdoc in France. Again, I have a question for Dheeraj. So, how does the system during the process of endocytosis, how does the DNA doesn't get degraded or something like that?
Yeah. Thanks. Yes. So if you have like these DNA are degraded by the enzyme called as exonucleases or the nucleases.
Sure. Yeah.
Now what we do, I did not show one slide, is we actually ligate those DNAs. So for the exonuclease to act on the DNA, it needs an open end, like 5' end, where it can bind and start degrading. So we ligate these these cages, So there are no open ends available for degradation.
Okay. So it's like a complete circular molecule.
Yeah, absolutely. You can say that. Yes, it's a catenated circular DNA. Yeah.
Okay. Thank you. A very interesting talk.
Yeah. My pleasure. Yeah.
Yeah. So, Dr. Bhatia, actually, I wanted to ask one question. So, like, can these cages be also used for like diagnostic applications? And like how?
Absolutely. Yes. So in fact, we are now we have this for patent work. What I did not show you was that, let's say if you make a 5-star based DNA, like a 5 DNA or a 5, it is like a star shaped DNA, not just cage, but any 2D material. And if you program it with the peptides, which can recognize the unfolded or slightly aggregated versions of the tau. So idea is to develop the sensor or in the very, very early events of the neurodegenerative diseases like the Alzheimer's. The problem with Alzheimer's and Parkinson's is the early diagnosis is a challenge. Like, can we now capture the very early events of the protein aggregation? Or even now we had one project for the COVID diagnosis where you can functionalize these DNA cages with single-stranded DNA, which can capture the RNA molecules putting the solution from the COVID.
Okay. Yeah, but
so these are like 2 or 3 areas where we are developing. Yeah.
Okay, that's just one more question. Like, so for like Alzheimer's, so this can will this be like a blood-based test or how can it be like?
Yeah. So idea is that maybe we'll need a small biopsies. Like we'll need a patient cell, but yes, we'll try. Like at this moment, blood-based test is a bit of a challenge in terms of neurotransmitter disease, because like these proteins are not much secreted. Like unless you purify the exosomes, it is difficult to develop the blood-based test for these ones.
Yeah. Yes, that is true.
Yeah, absolutely.
Yeah, and just one more thing.
That's a good suggestion. Thank you. I'll think about it. Yeah. Yes, please. Yeah.
Sorry, just one more thing. So like, so when you're doing these studies, so like for transferring this, like how do you deliver that DNA cages to the cells? Like with what transfection methods or
so
like?
These no transaction. So that is a big advantage. That there are actually receptors on the membrane which can bind to the negatively charged molecules. These are called as anionic ligand binding receptors, ALBRs. And thankfully now we know that they get clustered into the clathrin bed. So yeah, it gets internalized through the receptors. This is something which we did in the PhD. We have published it. In the C elegans, if you block those ALBRs, the endocytosis of the DNA cages is completely lost.
Oh, okay.
Yeah, and also other way to do it is use some other negatively charged molecule. Like for example, let's say I take a protein albumin and I modify the albumin with all the negatively charged molecules like myelylated PSA, and that protein will become negatively charged and it will act as a competitor for the DNA cage. And if you mix this negatively charged albumin with DNA, we compete out the DNA endocytosis.
Okay. Oh, yeah. Thank you, Dr. Bhatia. That was very interesting.
Yeah. Thanks. Yeah, my pleasure. Vikas, I think Vikas Verma.
Yeah, Dr.
He raised
Vikas.
a hand.
Yeah. Yes. Yes, Dr. Vikas, would you like to add? Or hello, we can't hear you, Dr. Vikas. Maybe you can type your question, Dr. Vikas.
Hello, can you hear me?
Yeah, now we can hear you.
Yeah, now we can hear you. Yeah, hi.
Thank you so much. Sorry. So Dr. Bhatia, my question is regarding your return back to India. So when did you decide it? Because if you just go join a postdoc and you say that I want to go back to India, does it affect your career prospects or the opportunities while doing your postdoc?
Absolutely not. No. Yeah. So the day your first big paper decides when you want to come back to India. If you go for a postdoc, let's say if you leave your country and when you're coming back to the country, the biggest question which every institute will ask you is, what have you achieved from your postdoc? So either, I'm not saying it's a nature or a science paper, but a good paper, a good publication from your either a publication or a patent or something which is which can be quantified. That is your major which decides. So when a typical time is 3 to 4 years, and when you have a good bunch of papers out, and that is which makes a profile really strong. So my postdoc, I had this Human Frontiers Fellowship and then this LabX. So at the end of 3.5 years, the first paper was out, and a co-author paper in Cell. And I thought, yeah, now it's good time that we can start applying. And remember, the day you start applying to the day you get offer and all, it is almost a year or more than a year. So during that time, you can kind of finish, the remaining publications or papers.
So what about the grants, while in postdoc, so you can gain some experience, because when you come back to India, you need to write.
Of course, yeah. So yeah, absolutely, yeah. You can apply this welcome test. When I was in Paris, Curie Institute, and my postdoc supervisor, he kind of gave me a lot of freedom. I was writing his grants, so I was helping him. So during doing that, I kind of learned how to write the grants and all. So this helped me when I was applying to India. Thankfully, the number of interview letters which came was pretty good. 10 out of 30 was a good number. Interviews in India. Of course, offers were very less, but no worries. Yeah.
No, it's very interesting talk.
Yeah, we'll be happy to discuss more. We'll be happy to discuss more if because.
Sure. Yeah. Thank you.
Arindam, I can see.
Yeah, thank you so much, Dr. Bhatia. And so I guess our next, so now our next speaker is Dr. Arindam Mondal. He's assistant professor at IIT Kharagpur. Also from IIT Kharagpur. And he'll be telling us about backbone viruses and their zoonotic transmission potential. Dr. Mondal?
Hello, am I audible?
Yeah.
Yeah, very good evening to everyone. It's a pleasure to be in the meeting. I first of all, I would like to congratulate the organizers for making this wonderful event, for organizing this wonderful event, and thank them for providing me the opportunity. It's been a while since I'm associated with this particular forum. Just how to actually, how to remove this panel.
Which panel? We can see it fine actually.
Fine. Is it fine? Okay. Okay,
Yeah.
not a problem. Yeah.
Yeah.
So I have been associated with this particular Sci-ROI forum for a long enough period of time, because I remember the first time I have presented in the Sci-ROI, it was the 2016 Chicago meeting, when we were in the panel of organizers at that time. So that was the first YIM meeting organized back in Chicago. But anyways, subsequently, I moved to India and joined the IIT Kharagpur. And today I'm going to talk about how we are trying to identify specific molecular signatures, which actually dictates the zoonotic transmission of bat-borne viruses. Now, first of all, what is the zoonotic transmission? Now, we have a lot of animals surrounding us, and all these animals have different kinds of deadly viruses, which in the usual course are not supposed to infect human. But then these viruses are smart enough to constantly introduce mutations in their genetic material, so that they can evolve and adapt into the human population, so that they can cause outbreaks in the humans. And this kind of cross-species transmission of animal viruses into human is known as zoonotic transmission events. Now, it is very important to understand the mechanism behind these viral evolution processes, because that's how we understand that how these viruses enter and replicate into the human host, as well as how it exploits different host factors in order to establish a successful infection within the human cells. And all these informations in a cumulative way help us to develop therapeutic as well as diagnostic strategies against these deadly human pathogens. So in the molecular biology lab, we are working almost in each and every aspect of the virology, starting from virus evolution to virus replication, host pathogen interaction, drug discovery, as well as diagnostics. And these are the main workforce who are contributing in these different projects. I'm going to talk about them later on, about these individual fellows. Okay, so today's topic is bad viruses. I think all of us know that bats are the natural reservoir for some of the deadliest human pathogens, deadliest human viruses. I am showing here three different classes, three different families of influenza of viruses, bat-borne viruses. One is the Henipa virus, which constitutes of the Nipah virus as well as the Hendra virus. Then the filovirus, which has Ebola, Hanta, as well as Marburg viruses in their family. And then the coronaviruses, obviously everybody knows about coronaviruses. They have the SARS, MERS, and the SARS-CoV-2. Now, all of these viruses causes minimal or no symptoms in bats, because bats are the natural reservoir for them. So this is the host with which they have equilibrated with. But then whenever these viruses cross the species barrier and gets introduced into the human, that's when they can cause severe epidemics or pandemics. And we have all encountered these kind of epidemics and pandemics during the course of history of viruses. Now, it becomes very difficult to understand, to study the evolution of bat-borne viruses and how they adopt into human population, because all of these families of viruses are extremely deadly pathogens, and you require at least the BSL-3, if not BSL-4, in order to culture or study them. In our lab, we are studying influenza viruses, and influenza, again, is a zoonotic virus. Now, let me tell you, wild aquatic birds are the natural reservoir for the influenza viruses. And from these birds, different kinds of influenza viruses cross the species barrier and then infects a wide variety of host species. I have denoted different variants of the HA and NA molecules, which usually cause infection in one particular host, so that you can see that how many different kinds of influenza viruses can be there in the nature. Now, different influenza viruses, as I was mentioning, constantly evolve to infect newer and newer hosts. Now, in recent times, only back in 2012 and 13, two specific subtypes of influenza viruses, H17N10 and H in bats, followed by which there was a major concern raised in the influenza virus research community about the zoonotic potential of these viruses, that is, whether these viruses can cause infections in humans. So in our lab, we have a major focus upon studying the evolutionary basis of these bat-borne influenza viruses and to study their zoonotic potential into the human and other host species. So if you look into the influenza viruses, it falls in the Orthomyxoviridae family. There are 4 different kinds of influenza viruses: influenza A, B, C, and D viruses, amongst which influenza A and B causes human infections. And again, among them, influenza A is the most, or maybe one of the most deadliest human pathogen across the world. So all of the varieties that I have shown you corresponds to the influenza A viruses. Now, influenza viruses are the RNA viruses which contains 8 different segments of their genomic RNA. Just like we have 23 pairs of chromosomes, influenza viruses 8 different RNA segments as their genome, which can encode up to 14 different viral proteins. And 2 of the viral proteins, the HA, the hemagglutinin, and the neuraminidase, actually determines the subtype of influenza viruses like H1N1, H3N2, etc. Now, if you look into the core of the virus, you'll be able to find these genomic RNA segments, but these RNA never retains as naked RNA. Rather, they remains or resides as ribonucleoprotein particles or viral RNPs. Now, what are the RNPs? RNPs are made up of the viral genomic RNA enwrapped with multiple copies of nucleoproteins in their oligomeric form to form these kind of double helical hairpin-like structures. And they also associate with the viral RNA-dependent RNA polymerase, which is the core replication machinery for the virus, and which is composed of the 3 different viral proteins, the PB1, PB2, and PA. Now, the RNPs are the main replication machinery of the influenza viruses, which carry out viral transcription as well as genome replication inside the cell. And the RNA polymerase is the major determinant of the viral species tropism. So in terms of species tropism, I'm trying to explain that it is the virus replication machinery which determines that whether virus can successfully replicate in a particular host and hence can cause infection in that in the respective host. So it has long been known that the PB2 subunit of the influenza virus polymerase actually is the major determinant of the species tropism. There is one particular residue, which is the 627th residue, which in all the bird influenza viruses remains as a glutamic acid, which is the negatively charged amino acid. Now, birds are the natural reservoir for the influenza viruses, and these bird influenza viruses are kind of attenuated or poorly replicating in the human host. So usually influenza viruses are not supposed to infect human. But then whenever we have isolated influenza viruses from human, it has a lysine, which is a positively charged residue at the same position. What does that mean? That means during the course of evolution, the virus RNA polymerase can adapt a specific mutation at the 627 position of the polymerase, and thereby which actually facilitates its replication in the human cells. In order to replicate this particular phenomenon in the in vitro setting, in the cellular setting, we have actually used one particular human adapted virus, which is the H1N1 WSN virus, WSN strain, which has been isolated back in 1933 and well studied. And if you just reconstitute the viral RNA polymerase by transfecting the individual RNA and protein components inside either the human or chicken cell, this human adapted virus can replicate well both in human as well as chicken cells. But then if you introduce the glutamic acid at the 627 position, that means you mutate the lysine with the glutamic acid, it becomes the bird type polymerase, and which becomes only functional only in the chicken cells, but not in the human cells, showing that bird virus polymerases remains restricted in human cell. And it was known for a long time. Professor Kanta Subbarao has shown it for the first time in 1993, but then it took about 23 years in order to understand why these bird virus polymerases remains restricted in the human cells. Only in recent times, from Wendy Buckley's laboratory from UK, they have identified a particular host factor, which is the ANP32A protein, which is responsible for the species-specific restriction of influenza virus polymerase. So they have shown, and the data has been recapitulated in our lab, where you can see that in absence of any ANP32A protein, the wild type polymerase, which is the human type polymerase, can replicate well in human cells, whereas the bird type polymerase is restricted. If you overexpress the human ANP32A, the restriction becomes even more prominent. But then if you overexpress the chicken ANP32A, which is significantly different from human ANP32A, you lost the phenotype. That means the bird type polymerase can replicate as good as the human type polymerase in the human cells. So what does this tells us? So avian influenza viruses, that means the bird type influenza viruses, always have a glutamic acid at the PB2627 position. And it is the chicken ANP32A which can support the activity of this particular polymerase, having a E at the 627 position. Human ANP32A cannot support this function, and that's why the human cells become non-permissive towards avian viruses. Now, whenever this avian type viruses have a mutation, the E627K mutation, subsequently it can gain functional activation in the human cells. So the bottom line of the story is basically a single amino acid change leads to the zoonotic transmission of the influenza viruses from birds to humans. Okay, that's all about transition or the transmission of the bird viruses into human. But what about the bat viruses that I have mentioned at the very early slide of my talk? So bat viruses, as I have mentioned, has only recently been identified, which raised a lot of concern that whether bat viruses can cause infections in humans or birds. And more recently, it has been shown that bat viruses are severely attenuated, significantly attenuated, in terms of their replication in human or bird cell lines. Okay, so we wanted to understand that why bat influenza viruses are attenuated in human cells. In order to do that, what we have done is basically we have performed an extensive bioinformatic analysis of the PB2 protein sequence from a wide variety of influenza viruses, starting from human infecting, bird infecting, swine infecting, etc., from all different host species, including bats. And what we have find that there is one particular residue, which is a molecular signature for these influenza viruses, this 282 residue. And that remains always a negatively charged residue like glutamic acid or aspartic acid. So if you see in this particular alignment, here we are showing all of the influenza A viruses, influenza C viruses, D and B viruses. You always have either the glutamic acid or the aspartic acid at this particular position. But then in the bat influenza viruses, you have a serine at this specific position. Not only that, if you look more closely into this particular residue, it is surrounded by two of the serine residue, which makes it a SES motif, which is again conserved across all different influenza A viruses, as well as influenza C viruses. Okay. And that particular motif remains surface exposed. And this ACS residues remain situated at 3 consecutive turns of alpha helix, which is completely surface exposed. Okay. Which is making it a unique surface exposed structural ACS motif, which is present in all of the classical influenza viruses. But then in case of the bat influenza viruses, that same motif becomes SST motif, lacking the central glutamic acid residue. So we wanted to check that whether this ACS motif is important in terms of the replication of the classical influenza viruses first. So what we did is, again, we have used this H1N1 polymerase activity assay. And we saw that if you just mutate this SES motif with a triple alanine residue, the activity of the polymerase is completely gone. But then if you even mutate only the central glutamic acid, the polymerase is about 70%, 60 to 70% effective. Interestingly, when we have introduced a bat-specific serine residue at the very central position of the motif, thereby recapitulating a SS motif, that polymerase is also defective. We have also generated completely bat type polymerase, which is having a SST motif, which is the characteristic of the bat type polymerase. And that too is significantly defective in human cells in comparison to the human virus polymerase. And I'm not going to get into all the detailed data that we have generated in order to support this hypothesis, but we have shown that if you introduce these mutations in the influenza virus, actually, we can generate recombinant influenza viruses in the lab. So we have generated recombinant viruses, and we have introduced this particular mutation in the human viruses, and that becomes attenuated. So based upon all those data, we have proposed that the bat influenza viruses replicates poorly in either birds or in the human cells because of this particular species specific restriction element, which is present at the 282 position, which is the serine. So based upon that, we can get a little bit comfort by saying that bat viruses in their current genetic form is less likely to cause a severe disease in humans. But the question remains that what would happen if the bat PB2 at the 282 position can have a species-specific mutation, thereby having a glutamic acid at the 282 position? What would happen? In order to test that, we have reconstituted the H17N10 polymerase, polymerase activity in the cell. And this is the H17N10 wild-type polymerase, which is having the SST motif, which is the characteristic of the bat-type polymerase. And then once we introduced a glutamic acid at the center of the motif, thereby recapitulating a human type polymerase that is having a SET motif, that becomes twice as active as the wild type bat polymerase. That means you can artificially, or through the course of the evolution, if you have this mutation in the bat polymerase, it can gain additional fitness advantage in order to replicate in human or bird cell line. So based upon that, we have identified this particular residue as a species-specific restriction element, which is present in the bat influenza virus polymerase, which is responsible for its restricted activity in the human or chicken cell lines. And this could also serve as an evolutionary element, evolutionary conserve element, which upon mutation can help the bat viruses to gain fitness advantage into the non-bat host species. We have also recapitulated this particular experiment in presence of the human or chicken NP32A in order to check that whether there is any correlation between the 627 phenotype and the 282 phenotype. And what we have shown that there is no relation between them. You cannot actually rescue the activity of the bat influenza virus polymerase, which is restricted in human cells, by overexpressing either human or the chicken cell lines. That means we have identified a completely new evolutionary conserved element which can serve as a species-specific restriction elements for the bat influenza viruses. So to conclude, we have identified, first of all, a SES motif in the classical influenza A viruses, which is extremely important for its activity, specifically the central 282 residue. Not only that, the bat influenza virus polymerase have a SST motif, which is responsible for its attenuated activity or the poor replication ability in the human cells. And we have also shown that by adaptation, species-specific adaptation at the 282 residue, it can have a better fitness in human or the avian cells. So this particular work has recently been accepted in Journal of Biology. So now, apart from this particular study, which is completely basic biology study, I would also like to focus upon a very small study that has been done, and which is completely a technology oriented study. And here basically in collaboration with one
Dr. Mondal, I just want to tell you that your presentation time is up. So maybe you can quickly wrap up.
Is it 15 minutes or is it 20 minutes?
It was 15 minutes. Yeah, it was 15 minutes. It is 15 minutes actually.
Yeah.
But
yeah. So I quickly wrapping up. So here I just want to show that we have generated a first indigenous rapid nucleic acid test. So for detecting the RNA virus infections, which has been implemented for the COVID, for detecting the COVID-19 infections. So here we are just isolating the swab from the patient samples and then resuspending into a lysis buffer, which can then be put into this particular machine where it can actually undergo a specific reaction leading to the change in the color. And then subsequently, it can capture the image and give you a final data output in terms of positive or negative infection. So the full from swab to result, entire procedure can occur within 45 minutes. And this is why we are calling it as a rapid nucleic acid test in comparison to the RT-PCR test, where you have to isolate RNA and then subsequently you have to perform the test, which usually takes a couple of days in order to get you the results. But this particular portable machine that we have generated in collaboration with the mechanical engineering department can perform the entire test in the point of care setting. So we have developed, so in collaboration with multiple company partners, we have different prototypes, starting from the laboratory prototype. Then we are using this particular engineering prototype for our field trials, which is going on inside the IIT Kharagpur campus nowadays. And we have already tested about like more than now, I think 2,000 samples in 2,000 live patient samples within the IIT Kharagpur campus. And this is the particular final commercial prototype, which is supposed to be launched maybe sometime in early 2022. Okay, and again, this particular technology has been published. We have published this particular technology and presented this technology. And with that, I'm going to acknowledge my collaborators. So I have been asked to talk about one particular topic. Rather talking about one particular topic, I have 3 different slides to talk about 3 different aspects of establishing one researcher as an independent researcher in India. And one of the very important aspect, the first aspect I feel is the collaboration. Because whenever we are working in an Indian ecosystem, Indian science ecosystem, if you are not in Indians of Science or not in NCBS, those one of the premier research institutes, you have a lot of restrictions in terms of academic, in terms of having infrastructural support and all those things. And that's why it's very important to collaborate with pupils all across India or maybe outside of India. We have been fortunate in order to establish collaboration with a lot of different labs, either within the IIT Kharagpur or outside of the campus or outside of the IIT Kharagpur, even outside India. So for example, the bat influenza virus part, for the bat influenza virus work, we have collaborated with Professor Wang Lingfa, which is a well-known virologist who are working with the bat-borne viruses. We are also collaborating with the University of South Florida in terms of developing one particular drug against the COVID-19 infection. And in this particular work, we are also collaborating with Devesh. Devesh is one of our core member of the Sci-ROI team, and this particular collaboration has been initiated through the Sci-ROI platform that I should mention actually. And then subsequently, we have brought in multiple different labs in order to participate in that. We are also constantly collaborating with companies, and we have been fortunate to transfer 4 of the technologies which has been licensed by different companies. And one of them is a basically multinational company, multiple US-based multinational company. And being in the IIT setting, which is the major technology-driven institute, even if you are interested in doing basic science research, you have to perform some kind of technology-oriented or application-oriented research, which will facilitate your progression in the IIT setting. Second thing is basically funding. I should mention that we remain fortunate to secure funding from multiple different extramural funding agencies. When I joined IIT Kharagpur, I received seed funding from IIT Kharagpur, but along with that, I have immediately received the Ramalingam Fellowship, which was an excellent funding for a newcomer. And with these 2 different fundings, it helped me to kickstart my lab in the IIT Kharagpur. Subsequently, I have received the Early Career Research Award from DST, and then the STARS Award from the MHRD, which helped us to keep on running. During the COVID-19 era, we kept on working on and contributing in the COVID research. And as a result of that, we received generous funding from the IIT Foundation USA, which is basically a IIT alumni-run non-profit organization. Also, we have received ICMR funding in terms of against a call for COVID task force. And we are part of the Center of Excellence in the IIT Kharagpur, sponsored by the ICMR. And definitely intermittent industrial fundings helped us to carry out the application-oriented research. Last but not the least, the workforce. It is very important to have dedicated, hardworking people in the lab. So we have 2 different postdocs working, leading 2 different major projects in the lab. Along with that, we have 4 different PhD students. The particular batch story that I have shown is driven by Choptorshi, one of the senior most PhD student, all set to go out. And definitely, being in the IT system, we get a lot of internship students. We thank to all of our alumni who are almost all of them are internship students who contributed to different projects during different time points. So it is actually important to have these internship students. If you are getting internship students, don't consider them as a burden, rather try to motivate them so that they can contribute to the overall growth of the lab. With that, I would stop and then I'd be happy to take any questions if there are. Thank you very much.
Thank you so much, Dr. Mondal. That was a very nice talk. And thank you so much for sharing your journey also. And yeah, so I would ask the audience members if they would like to ask any questions. Kindly do so. Okay, then maybe I can ask one question. So, which is like a general question. So, like, what do you think that because there was like a big debate over whether bats were responsible for the coronavirus to get transferred to humans and causing the pandemic. What do you think? Because you said that it is not possible, for the bats to transfer in humans without the mutation.
So yes, so that is obvious. So basically, for SARS and it has already been established that the SARS coronavirus, which has caused the pandemic back in 2002-3, that was a bat-borne virus. And based upon the similarity in the sequence of the SARS-CoV-2 and SARS-CoV, we can definitely say that it's a bat-borne virus. But definitely, it has adapted more. Okay. So it might be an adapted version of the SARS coronavirus, which has caused a previous pandemic. Now, if you think about the overall pathogenicity or the virulence of the SARS coronavirus, it was way more severe than the SARS-CoV-2. And possibly that is why that particular virus was not very successful in terms of spreading across the world. So the more successful the virus, the more virulent the virus would be, it will remain isolated in a smaller population. And the more milder it would become, the more the chance for the virus to spread into wider population. For example, if you think about even the SARS-CoV-2, it is constantly changing. And the newer variants are even milder than the earlier variants, right? So that I think that's what you are asking. Right? Definitely, there are mutations that has been adopted by this particular virus, but in terms of the origin, I think it is well established that it is from the bats originally, although the intermediate species has not been characterized properly yet.
Yeah. Thank you. And like there is one question from Sachin Patra. Do humans also have natural viral reserve?
So for the humans, we do not call them as a natural virus reserve, but there are human viruses. Okay, so for example, there are certain coronaviruses which causes infection in human, and they are equilibrated within the human host for long enough period of time. Again, leave about coronaviruses. If you think about the polio viruses, or maybe herpes viruses, for example, the varicella zoster virus, you don't see chickenpox happening in other animals. So that's an exclusive human virus, okay, which has been equilibrated with the human for long enough period of time, so that now we designate them as human viruses, not as zoonotic viruses. Zoonotic viruses are those viruses which has recently been introduced into the human population. and that's why they are more deadly in nature.
Oh, there is. Okay. There is someone asking if they, Mr. Asif Islam is asking if they can apply for internship under you, but I guess he can ask you separately.
Definitely, just write emails. Write
Yeah,
emails.
you can. Yeah, you can email him,
Yeah.
Asif. Yeah, thank you so
Yeah.
much, Dr. Model, for this interesting talk.
Thank you. Thank you.
Thank you. So yeah, so now we have Dr. Someshwar R. Saguruthi. He is assistant professor at Department of Genetics, Osmania University, Hyderabad. So yeah, hello, Dr. Saguruthi.
Hi, ma'am. Can you hear me?
Yes, I can hear you. Yeah.
So, can I go ahead and start, ma'am? Richa, ma'am?
Yes, sure. You can call me Richa. Yes, sure.
Okay, so all of you can see my slides now. Is it visible?
Yes. Yeah.
Okay. Yeah. First of all, I want to thank the Sci-ROI for giving the opportunity to share my experiences at Osmania University. Okay, so this is, I feel that a privilege to share my experiences, what is happening within my lab and in my whole career. So I'll be sharing. Okay, so here, first of all, I don't want to take questions at the end. I thought I'm going to share a story, what is happening and how I shifted from US to here and what is my career. It's like a story only. Okay. So by that, I thought this is a STEM presentation. Most of the other biological students will be coming. That's what I thought, and I made a very general presentation, which will be pretty easy to understand each and everyone. Okay. So here, after me shifting from the US, everybody used to ask me, why state university? Why not central universities? Or first of all, why did you come? Okay, so at the end, I'll tell you the reasons why I'm here in the state university. Why? Because I belongs to from the Telangana state, and that's the reason here I am. Okay, so before that one, I just want to put up the other perspective. Okay, being in the state university is not always a bad idea, and we can also have positives. There will be a number of negative points will be there, so everyone knows any common civilian if we call, or any minimal educated person if we call. So no state university is in India at least. Okay, so it's not very not well known for the research or the progress. Okay, so irrespective of that one, I joined this university. Basically all the universities in India, and most of the Osmania University, which is a 100 years old university, which is known for the some background noise is coming. I'm audible for all of you?
Yeah. Yes, there is some noise. Yeah.
Okay. So this is the 100 years old university, which is known for the art and expression. What's
So
happening, ma'am? Is there any?
It's Miss Shireen. I think Miss Shireen can mute. Can you please mute your Miss Shireen can mute, please? Yeah, thank you.
Yeah. Okay. So this here I am. So this university gives the freedom of thought and expression. Here there is no any you don't need to stick to the one specific field, or you can have a number of collaborations. You don't need to have a high-end permissions from the director or your mentor. So here you have freedom to choose your own projects and progress, and that's the freedom I really enjoyed. Okay, so that freedom comes with some of the challenges also, but being in Osmania University or any state university, so that freedom is really good to enjoy, and you can progress in the way you want to take your research, you can take it up. So that's what I'll tell all the abroad PDFs or the other research scholars. So it's not bad idea to join the state university. Like if you have a willing to take the challenges and if you're able to face the problems in the local place, so you can come and join the state university. Still, you can be successful also. That's what I feel that with the challenging conditions have been published a number of papers and they have been successful. Okay, so today's topic basically I'm going to focus it on insights into the inhibition of peptoglycan pentaglycine cross bridge, forming a FEM proteins of methicillin resistant staphylococcus aureus. Okay, so here before coming to that one, as I told, like here I have a education from the, like I did my master's from JNU New Delhi and also PG diploma in bioinformatics. Okay, why? Because I'm sharing all these things is like all these experiences, what are my background I have, that really helped me what I've been doing now after joining the Usman University. Okay, so and I did my first PDF in the San Diego TSRI, then second PDF with the Nobel Foundation in the Ardmore, Oklahoma. So then I took a regular appointment in 2013 as assistant professor in the Department of Genetics and Biotechnology. And here also you can see this one. This is my personal mail ID. If you have any doubts or the scientific point of view or anything, so you can mail to me or you can ask me right now also. And here I'm just going to give the overview of one of the topics. And if you need to have more about, if you want to know more about my research, what I've been doing in the Department of Genetics and Biotechnology, you can view my personal lab page, which will give the more insights, and my publications are there. So where you'll get a more scientific version of the things. Okay, so here, like I was trained as a protein crystallographer. I did my PhD with the Professor Manad Murthy, who is known as a virus Murthy. So he is the person who is carried out structural virology work in India, and he is the person who determined the Physalis mortal virus and Suspenia mosaic virus. And I've been I had a training as a PG diploma in bioinformatics, which really excited me. Like after having a biology background, learning computers to use that one, and so that is the way I took that path. And I feel that at the end of this one, it gave me a lot of fruitful results in my PhD days, as well as in the my own research lab when I started, as well as in the PDF also have most of the things on computer, like a cakewalk I used to carry out in the structural biology works. Okay, so these are the things during my PDF and my PhD days. As I told, I was trained as a protein crystallographer. I could determine the 19 PDB entries and I could submit into the protein data bank. So it's part of my PhD days where we determine the mannose phosphatase place. So this protein crystallography actually gives the spatial arrangement of each atoms, how they're placed into the and how they're coordinated, how they're proteins are the end things, like what are the biological information will be there in the DNA. So those will be performed in the form of the proteins only. So they will try to accomplish the functions or the information whatever is there in the DNA. So those proteins will try to carry out. There's maybe the structural form, or there's maybe in the enzymatic form, or so different ways and means they try to perform. So basically by using this crystallography work or structural biology work, so with the inhibitors and different things, we proposed a probable isolation mechanisms in the these things. Okay, so this is a part of my PhD work which we carried out in the IISc Bangalore. So later I had a major projects in the Scripps as well as in the Nobel Foundation. So that will take a long story. So basically I try to focus on what is happening in the me in my lab. Okay, so when I started as a principal investigator, so I had started with the short-term goals as well as some long-term goals. So I used to asking simple and answerable questions. And why? Because I have always minimal setup is there. So in this one, so to be successful, we had to ask the questions which can be answered, and as well as not to be compromised with the asking challenging questions with the long-term goals. So I started my lab. Being in the state universities, the main role is to being a teacher or the educator, and so that is the major component of the time it will be taking up. So because of that one, will not be eligible for the Wellcome Trust grants. Why? Because they ask you to write the 80%. You have to give the 80% of your time for the research. So there some demerits will be there, but here when you are in the state university, like when you're teaching, you learn many more things, and every day you'll be up to the date. Okay, so here in the short-term goals, I used to write this startup grants and setting up the lab and guiding students. So when I joined the Usmania Department of Genetics, so just they gave me 4 walls and some lab. Okay, so from that one, with the generating some of the money from the different labs, I tried to build up one state-of-the-art facility in the Department of Genetics, that will be in the state universities. Which, like, my lab is almost well equipped, as good as the DBT or the CSR institutes. Most of the things which are necessary for my proposed work, we could equip in my own lab. Okay, so here, like, have a molecular medicine lab. I named it as well because as from the structural biology group, and so most of my focus is to design the drugs and to identify the. So one more other aspect is like a chemistry department. Usmania is one of the strongest department. Almost 30 to 40 faculties are there who works in the chemistry department. So had a long-term collaborations with those people, many young and productive researchers from the chemistry department, and they also we usually share the different aspects, and that also really helped me in the to be successful in the department in the Usmania University. Okay, so here also, like, asking the challenging questions, like not only the simple projects and asking the human antibody, like this is the larger perspective and many more things also we have. Human antibody production plants with the lower price, or with the any other thing, like I want to produce the antibodies with the lower price, by that any common civilian, the poor people can also use these antibodies for their different treatments. Right? Because so these antibodies, when we see this one, they'll be having a very specific activity. They can stop it, that one, whereas the other drug molecules, they'll come with the several side effects, and they'll not have a very successful when to come into the market, or the many things, like a very market any drug molecule what we synthesize in the laboratory or in the as a pilot project to come into the market will take a longer time, and that we will have a lot of clinical trials and many things. They'll try to end up in the other side of the story. But whereas the human antibodies, like those will be having a very specific activity. So that's why, like I thought of it has antibody production will having a lot of focus. So that is the major goal I had, that one. So that is the challenging question which I'm trying to focus in the longer term goal. Okay, so here in the Usmania Department of Genetics, like based on my background, everything. So any project which I start this one, most of the things, like we try to carry out the bioinformatics work, like that maybe with the genome-wide analysis with the MTB or the equali, what are the genes are there. So first of all, we try to spend 3 to 6 months in each project and the different bioinformatics analysis. So that's what is my lab is. My lab scholars are the most of my lab is like a hybrid lab. So almost 30 to 40% of the work we try to do it in the computer, and rest of the 60% work in the laboratory. So both way, like this is the best way to train the graduates, and that's what I've been doing, and which is giving a very fruitful results. So here in my laboratory, usually we'll have a bioinformatics, so genes we try to identify which have a specific function, or which is vital or essential for their function, and which can be inhibited or for the upregulated, downregulated, many things we try to characterize that particular gene for their its activity. So once the bioinformatics analysis has been done, so then I try to take that particular gene and try to amplify the genes and try to clone that one, and try to do the expression and try to confirm the cloning. So this is from the bioinformatics lab to the bring it to the molecular biology lab and try to do the small scale expression and the pilot scale expression, try to do it, then try to optimize. So many times, so what I'm showing here, things will not be so easy. Some of my scholars will get frustrated in the cloning as well as expression and solubility. So whereas some people, they try to get some tons of protein, like not in the tons of protein, like mg protein, whereas some other people, they try to get into the very largest, very small amount of the proteins. So they had to optimize from the different ways and parameters will be there. So we try to do that one. Then large scale production and purification of these proteins. So this is the training which usually I give it in my laboratory, and which is having a lot of job opportunities in the outsiders. So this way I've been trying to do it into my lab. And once I started into the Usmania, like I also have a very story. Like, so when I write to any granting agency, so when I used to write the very high-end projects, then they used to think that I cannot carry out those proposal projects in Usmania University, and they used to reject. So if I write a simple project, then they'll try to think that there is no novelty in this project and it is very simple. So this type of problems as will be there. So then because of the time I could publish the papers, and so with my publications being in from the state university, when I've shown that one, so they got a confidence and they're continuously getting a different, I'm attracting the funding from the different funding agencies. Like most of the great goods, the SERV people, SERV has been a major backbone for my research lab, and then DBT, BIRAC, and UGC. So even recently we also got from the TS cost also some of the project from the state scientific community funding agencies. So these are the things I've been trying to do this one. Like here, like a major thrust area of my short-term goals is the working on the antimicrobial resistance. So as I told, we have a strong collaborations from the chemistry department, and so they have been publishing synthesizing several compounds, and which is giving also. So as part of this one, so 7 lakh people die due to the antimicrobial drug resistance resistant diseases, and almost 24 million people to extreme poverty because of this antimicrobial resistance. So this is one of the unanswered questions still as we are evolving. Even this microorganisms has been evolving with us also. Okay, so as we are keep on developing the drugs, even they are also keep on developing the resistance, and many ways and means they have been escaping the drug molecules. So there is no end for this particular field. So I thought I tried to focus just one branch of the main focus of my research lab on the antimicrobial resistance. Okay, so as part of this one, like we have a cell wall and a DNA, RNA synthesis, the folate synthesis, so cell membrane, so the protein inhibitors, nucleic acid inhibitors, or cell one inhibitors. So many targets we can have it, and even this microorganisms also keep on evolving and developing a antibiotic resistance because of inactivating the enzymes like beta lactams, or the aminoglycosides, or the macroslides, and the rifams. So many things are there. So they try to, what are the drug molecule we're designing? So they have been inactivating the particular target activity, and they are escaping this multi-drug resistance they have been acquiring. Okay, so target modifications sometimes they're happening at the immunity bypass, or so these things are happening. So there is still a number of unanswered questions are there which has to be explored. So with that one, I took it up that project as we carried out the MRSA superbug. So this is a methicillin resistant staph aureus, so which is considered as a superbug, and which is caused most of the hospital infections. So which is not it doesn't cause a deadly infection, but if you're immunocompromised or if you have a comorbidity, so then it will cause the lot of severe things you have to face it up this one. Okay, so we come to know that when this is escaping the most of the drugs, and which is having which is considered as a superbug, and we try to analyze this is the initial project. We try to analyze this staph aureus genome, and what has been done, and what is unanswered things are there, where is the gaps are there. We try to analyze this one and why it's happening. And so when we found this one is a MEC gene, is the carries a MEC gene, and which encodes a alternative to non-native penicillin binding proteins are there. So this is called the PB2A. So this is only specific protein MEC gene, and which codes for the different set of proteins which is not present in the other pathogenic organisms. So then we thought that what is the still why we are not able to design an appropriate drug. So we carried out the different analysis and found that it codes for the low affinity binding beta lactams and functions as a native PBs. Okay, so this one also has a special peptide glycan cross-linking which is present in the MRSA. When we analyze that genome-wide analysis, it has a very specific protein known as a FEM proteins, whose structures are only one FEMA, one protein is determined at the time, and there are 3 more proteins are there which is structures are not available, and not much work has been carried out. So then we try to study these proteins, what they have been doing on the MRSA, and how they're playing into the role in the escaping the antibiotics. Okay, so here when we found that one this FEM proteins also in the expansion we try to call this factor essential for the methicillin resistance, and so this have a ABC and X proteins are there. Okay, so basically they'll have a femtag peptidoglycan will be pentaglycine bridge to they'll form in the cell wall. Okay, so here the FEMX is they provides the first glycine, and FEMA forms the second and third glycine, and FemB fourth and fifth glycine, and FemC is the last one emanation of the peptidoglycan and glutamate. So all this, whereas the only one protein structure was available at that time, and the rest of the protein structures were not available, and not much focus hasn't been done. Okay, so when you see the most of the bacterial system, they'll have this one. So most of these Muria, these protein structures are crystal structures are available. So crystal structures are essential for the designing of drugs. Like most of the drugs, we try to do it with the structure-based drug designing, and based on that, we'll have a we can identify the active site, and where we can design inhibitors, and exactly they're fitting into the cavity or not. We can So all this information, Muri, all these protein structures are available, except this FemB and this FemX, and so other protein structures are not available. So then we thought that this is the one of the best bets to focus it on. So basically, so this is the pentaglycine. So when this bridge is not allowed to form, so immediately any one of the drugs can work on this one, and their cell wall will collapse, and we can kill them. So that's what is my main objective. We analyze this one. So these proteins, what are the pentaglycines which are forming this one? So we targeted all these proteins, and so which basically, so this is the gene complex and FemABC gene operon, and so basically, this structure is available. So this FemX and FemB and FemC, whose structures are not available, and formation of interpeptide pentaglycine formation, they have been playing a role. So basically, like what we've seen, like here we can see this one. So this is the pentaglycine. So when this pentaglycine is not formed, so then they're prone for the they cannot survive, and they cannot go for this one. Okay, so here basically pathways involved in cell growth and development. So these Fem proteins, basically they play a role in the cell wall synthesis. So we, with the work with our lab people, we cloned these 3 proteins and FemB, FemC proteins in the PET vector, which we regularly use it as expression vectors, and we confirm their sequences, and so we could express these proteins into the 3 of the proteins in the BL21, which had all of them had come into the soluble proteins, and we are trying the crystallizations. So here in our lab, like basically we try to use the nickel NTA affinity column protein purification, and we try to purify these proteins at the large scale, and we try to check into the is it the single banded come or not. So for the crystallography work, anything we need to have a very pure and homogeneous proteins. So I'm not explaining all the insights of this technical work. I just trying to show this one. Okay, so when we get these purified proteins, like then we go for the crystallizations. So we use it to purify these proteins by using the active purifier, by using size extraction column chromatography, where we get it into the pure proteins, and where are the impurities are there, we try to avoid those proteins. So then we go for the dynamic light scattering, which is like tells that this protein is homogeneous, or is there any contaminations are there, is it so basically like this polydispersity PD% is there. So what is the peak? First one is there. So this is showing into the 115 Dalton. Okay, and intensity. So this is polydispersity. If it's less than 15, so then there is a more probability that this purified protein will go for the that can be crystallized. So then lattice easily can be formed. So with that idea, so before going for the crystallization, we try to do some of the biophysical characterizations as part of that one. So we do the dynamic light scattering, and as well as the before ITC and any other studies, we try to do it. Then we try to go for the crystallization studies. Okay. So based on that one, we try to get into
Dr. Sagurate, sorry to interrupt, but your time is almost up for the talk. So maybe you can, yeah, you can wrap it up quickly.
It's almost over. Yeah. So as part of this one, like we try to do the crystallization studies, and so this is like a very competitive world. Like when we started this project, same project has been initiated in the UK also. Those people submitted this FemB protein structure. Okay, so they have been determined this structure, and so they have a much more advanced facilities are there. And due to Corona, we couldn't get into the in time. So then we try to do what are the structure they determined. So only FemB is there. So now I'm focusing on the FemX and FemC proteins. I've been trying to crystallize those proteins, and we have been trying to do this one. So along with this one, like people like we try to do some of the inhibitor synthesize the compounds with the collaboration with some of the chemistry department faculty, and we published some of the papers with the general molecular structures and BMCL. So a few more papers we have been published. And during this course of study, also we published the evolution of next generation beta lactams in the JSM biology and BMCL. So most of these small molecule studies have been publishing in the different journals, and these are the things we try to be publishing into the several reputed journals from the Osmani University itself. Okay, and so we also, even though this is a state university, we also have a state-of-the-art facility for the most of the protein works, and I built a microbiology lab, and which is having most of the facilities for the protein purification of the things. And I also have developed the bioinformatics lab, so where I teach the master students as well as my use for my research purpose also. And these are my research scholars who regularly come to the lab, and so all credit goes to them. So what are the work and results we got it. And along with this one, so this is my end of this talk. And I also have some more other publications we published on the asthma, which is published in the collaboration with some of the things. Maybe whenever I get a time, I'll try to share this one, so you can find more insights into my other asthma work also. Here, this story is like a asthma patients. Whenever we go to the doctor, they usually prescribe the salbutamol. But some of the patients, if it works, that will good. But some of the patients, almost this project was in collaboration with the chest hospital we carried out. So some patients will come back. So then doctor used it to prescribe the Finitrol. So then it used to work. So Dr. Srinivas asked me what is happening. So then we seen that the patients who is not working. So we collected those blood samples and we analyzed this ADBR gene, and we found that they have this 3 into isoleucine modification usually happen. So those patients, the salbutamol will not bind at the active site. So those people are having a better activity with Finitrol. So this particular thing also we published in the gene paper. Then we also have a current pharmaceutical design and many other publications that come up. And with the other work also, this is my recent one of my student submitted work on this allogeneic base inhibitors. So this was published in the EGMC, European Journal of Medicinal Chemistry, which is having a 6.5 impact paper. So this was a corresponding author as the first paper, which we published, major paper from my lab. And here, when we study this one, there is a active site flexibility used to be there. So based on that one, we designed a new bigger molecules and which can fit into the active site, and we shown that is working better than the already existing molecules. And I also thanks to granting agencies, UGC, SERP, and DBT, and TS Cost, and many other small contributions that come from the different agencies and some of the companies also. So if you have any doubts, I would like to take it and answer questions. Is it okay, Richa ma'am? Got it?
Yes,
I'm in the
yes.
time?
Yes, yes.
Thanks.
Thank you so much, Dr. Sagaroti, for sharing your work and experience. So yeah, so if anybody has any questions, they can ask now.
I think it's pretty clear. I think crystal clear. So that's why they don't have any.
You explained quite much. Okay, then if nobody has any questions, then I would like
any questions. If you're hesitant to ask here, so you can post the questions to my mail ID. I'll be sitting from 9 to 5 o'clock in Osmani University. Anytime you can walk in. You don't need to take permission from anybody. So Osmani is like anybody's place. So just have to walk in.
Yeah, thank you so much, Dr. Sagaroti. And then I would like to thank all the speakers. And I hope everyone got to learn so much here from your experiences and from your work. Yeah, so thanks so much again. And then I would like to end the session here.